Hi, I'm Ailey. So thanks for joining us on today's webinar, Bridging Genomics and Proteomics for Next Level Immunopeptidomics Discovery. So for today's topic, we will focus on the new features in our PIX 12.5 peptidome workflow. Here's a quick overview of what we will cover in this talk. So we will start with a brief introduction to immunopeptidomics to give you some background on this important research field. And next, we will dive into the new features of the DeepNovo Peptidome workflow, which is a built-in workflow for PIX Studio specifically designed for immunopeptidome research. So in the new features, we have improved our de novo sequencing capabilities with the GraphNovo algorithm, and we have group-specific FTR search features is designed to enable better control over the search space. And we also added a powerful new tab that allows you to visualize the origin of the identified peptides, directly linking them to their corresponding genes. So, what are immunopeptides? Immunopeptides are MHC-bound peptides that are presented to immune system. In class 1 pathway, endogenous antigens will be digested into peptides by proteosomes and transported into ER. And within ER, these peptides then associate with MHC molecules. If the complex is stable, then the complex is transported to membrane for interaction with the T cells. So there are endogenous antigens can arise from various alternations, including changes in genomics, epigenomics, alternative splicing, or translation of non-canonical regions. So then why are we interested in immunopeptidomes? These endogenous antigens can serve as a potential targets for cancer immunotherapy, offering a way to use the immune system to recognize and eliminate the tumor cells. So there are different cancer antigen sources. Within the cancer cell-derived antigens, we have tumor-associated antigens, TAAs, and tumor-specific antigens, TSAs. TAAs are antigens that are normally expressed in healthy cells, but are overexpressed in tumor cells. TSAs are mutated or aberrantly expressed peptides derived specifically from tumors. And TSAs are unique to cancer cells. So both types of TAA and TSAs can serve as a target for antigen-based cancer therapy. However, while TAAs may carry the risk of on-target off-tumor effects, where normal cells expressing the antigen could be also affected, TSAs are often considered to be a more attractive option for cancer therapy due to their cancer-specific nature. So there are several approaches for discovering new antigens. Currently, we have the two main methods for used are NGS-based identification and mass spectrometry-based identification. So in the NGS-based approaches, the process starts with sequencing the genomics, DNA, of tumor cells, and whole genome sequencing is performed to identify mutation in the tumor genome, including point mutation, insertion, deletion. The identified mutation are then in silico translated into peptides. The next step involves the prediction of which of these peptides are likely to bind to specific MHC molecules. And after the prediction, the peptides are ranked and prioritized based on their likelihood of being presented to the cell surface and recognized by the immune system. So the mass spectrometry-based identification of new antigens is more direct experimental approach. It involves isolating and analyzing peptides that are actually presented by MHC molecules on the surface of tumor cells. The isolated peptides then analyzed using mass spectrometry, and this method provides direct experimental evidence of the actual neoantigens being presented by the MHC molecules. So here is the comparison between different methods in antigen discovery. With the sequencing-based method, they are faster, and I would say they are cost-effective for large-scale screening, and they can provide a broad overview of potential new antigens across the entire genome. Limitations are they can be a sequencing error, and then it results depend on the sequencing accuracy with high false positive rate. With the sequencing plus mass spec search method, acquired mass spec data is searched against in silico database generated by NGS sequencing. But with this method, after our estimation would be issued with large search space. Another method would be directly mass spec based. With the mass spec based method, we can directly identify peptides presented by MHC molecules and by de novo sequencing with our method, without the need of database providing more accurate and experimental data on new antigens. The limitations on this method will be detecting of low abundance peptide. So more sensitive instruments and experimental procedures are required for the mass spec based method. So let's go into the project setup view on the PIX 12.5. So in the first step, the raw data files can be, after uploading the project raw data files, we can select the enzymes, instruments, and fragmentation method and acquisition method according to your own experiments. So for the immunopeptidomics research, enzyme would be none here. And for this example files, we use Orbitrap, Lumos, and fragmentation methods SCD, and acquisition is DDA. Right after this, in setup view, you will go directly into the workflow selection. Very straightforward. So for the immunopeptidome, you will have this tab. So you will just click that. And then within this workflow, we select error tolerance according to your instrument and PTMs according to your experiment. And then we select our target database here, human reviewed, and we have another new feature here. We added a non-canonical database, so you can have your personalized database added here. And if you would like to add a contamination database, you can also add the contamination database here. And the search peptide length can be set here for your preference. For HLA1, we usually set 7 to 20 length in length. So after this setting, we also have a LFQ setup view for comparison between the samples, and the QC tab if you all would like to see a quality control result. So that's all for the setup of the immunopeptidome tab of the PIX 12.5. So let's go into more details about how the search is done within this workflow. So peptidome workflow is specialized for peptidome data that combines the database search, both DB database, canonical database search and non-canonical database search, and de novo sequencing and the identification of the mutated peptides. So we train our deep learning models using peptidomics data to improve the sensitivity of peptide identification. So in this round of search, DeepNovo peptides and all the DB peptides and the spider search mutated peptides are combined for accurate FTR estimation. So we will have another round of FTR estimation here, and the outputted peptides is characterized as database search, DeepNovo database search, canonical peptide database search, non-canonical peptides, and mutated or homolog peptides and DeepNovo peptides. And the results can directly go into for like immunogenicity prediction or length distribution. Or if you're interested between the peptides distribution between the different samples, you can also do LFQ and quality control. So so we said that our advantages of our software would be the DeepNovo sequencing. So to correctly identify new antigens or non-canonical peptides with mass spec-based method, we really need to have a good de novo sequencing algorithm. So with our Pix 12.5, we use GraphNovo, which is our latest deep learning algorithm for DeepNovo peptide sequencing. So if in this sequence, if you name the sequence before the missing fragmentation region as before, after the missing fragmentation region as after, you can see from this bar chart that amino acid recall percentiles within the missing fragmentation regions are low. But you can also see that with the older models, the light yellow or light green bar chart, you can see that the amino acid recall percentiles are decreased, actually got affected for the sequences after missing fragmentation region as well. So, but with our path searcher guided GraphNovo algorithm, we resolve this issue. So we don't have this accumulated accumulated de novo prediction errors anymore. So we also tested peptide recall performance of all models with different number of missing fragmentation. And you can see here our latest algorithm GraphNovo performs best among all of them. So now we have this de novo sequences. So how do we evaluate our quality or quality control our de novo peptides? So these features are also in our Pix 11. So I will just repeat it again here. So we have retention time prediction, delta RT, and we also have the MS2 correlation numbers. And the mirror plot is shown here, comparing the measured spectrum of the peptide and the predicted spectrum of this peptide for further sequencing accuracy assurance. And each fragment ion mass to charge ratio and the intensity are listed for further clarity and sequence accuracy assurance. So beside from retention time prediction and MS2 correlation, we also have confident amino acid scoring here, which depends on the existence of both Y ion and B ion for the residues and the intensity of the fragment ion signals. So the residues with black means the lack of good fragmentation ions. We also have a filter to change the non-confident amino acids, which the black amino acids here, to mass tag for better clarity of the identification of the peptides. So another new feature we have with 12.5 is personalized training model. It can perform personalized training and scoring based on the allele-specific peptides for improved identification sensitivity. But this is not included in the installing package. So if you need this, you will need to email our support team for getting the installation package for this module. Another feature we have with Studio, I actually briefly mentioned this before, is the new non-canonical database search is incorporated our workflow. So so you can put your personalized FASTA files that you got from the NGS sequencing data and go into second round of search, where in second round of search, we will have FTR estimation for both non-canonical database and the target the canonical database, and give you the FTR controlled and sequencing results. We also have the validation only tab here, where you can align the non-identified non-canonical peptides to your non-canonical database. But we recommend using the second round search with this feature. So this is a case study on where they use they have generated in silico translated peptides from non-canonical circulic RNA and concatenated the peptide sequence to the canonical database and perform HLA purification and search the result against this concatenated database. So with their raw data and provided database, we also run this project, and with PIX 12.5 found most of the peptides in their publication and even more peptides that they did not identify. So this also applies for the non-canonical peptides. So PIX 12.5 identified most of the peptides in their publication, and we have identified even more non-canonical peptides they didn't identify in their publications. So in the gene tab feature, we can now map the identified peptide to the chromosome loci origin for visually better understanding the peptide biosynthesis. So in this view, you can see that you will, if you have multiple samples, here we have a tumor sample and benign sample, you can see that this canonical peptide is presented in both of the samples, and we map this canonical peptide to its gene origin with the chromosome 12 coordinates between this number and this number. And we also have this coverage information on specific gene, also how many peptides are identified or can be mapped to this chromosome. Within this view, so if you collect that specific peptide here, if you collect this, the spectrum view will show up for better viewing of the spectra or peptide quality. So we provide you this mirror plot, and you can see if the peptides in this mapped view is actually confident or not confident. We have the same peptide spectrum view for non-canonical peptides as well. So here, if you click this gray bar, the spectrum view will show up and then give you more clearance on how confident of this peptide sequence identification from this spectrum. So beside from our PIX 12.5 studio, I'm not sure if you know that we are now also doing this lab service. So in our lab service, we can do end-to-end preparation for your samples. So we can get your samples, and we will run your samples we will do the immunoprecipitation purification of the MHC complexes and run the purified peptides in our instruments, do the analysis for you, and upon that we also do the DeepSelf immunogenicity prediction for you and generate a report for you. So within this lab service, with your data, we also provide all the length distribution and binding affinity prediction, allele specific and binding affinity prediction, and the motif generation. Also, if you have multiple samples and you would like to see the quantification, we also do the quantification for you. And this is the DeepSelf, a personalized model for immunogenicity. So this model is also will be applied if you do the lab service, and this model is developed by learning how our T-cell central tolerance system works. We have talked about this model in our previous webinar, so if you're interested, you can see or watch the previous webinars as well, or you can email our support team. So in a summary, so DeepNovo Peptidome Workflow is a complete workflow for immunopeptidome research, and we have this GraphNovo algorithm, and it resolved the issue of accumulated prediction errors due to missing fragment ions. And we now have the group-specific FTR control approach to increase the identification confidence for both non-canonical or canonical database. And we have this personalized HLA peptide training, help to increase sensitivity and accuracy for identifying peptides. And we have this all new gene tab to understand the peptide synthesis or visually help you to map your peptide to the gene origin. So I think this will be all for today's webinar. If you have question, you can post it in the chat or you can ask me directly.